Clinical bacteriology laboratories equipped to diagnose bacterial infections and perform antibiotic susceptibility testing are scarce in low-resource settings. Correct identification of bacterial infections are necessary for appropriate antibiotic treatments. The Microscan walkaway is an automated benchtop analyzer that uses biochemical reactions to identify bacteria down to a species level. The main components of the walkaway system are the walkaway instrument, the LabPro computer, a report printer, and a barcode printer. Operating the walkaway instrument requires close coordination with LabPro. From initial instrument startup, you must interact with the walkaway instrument via LabPro to perform such functions as maintenance, loading and unloading panels, and monitoring panel processing. Historically, microorganisms have been identified by what are now referred to as conventional procedure, which include reaction in tube media and observation of physical characteristics such as collagen morphology, hemolytic patterns, and odor, coupled with results of gram staining, agglutination tests, and biochemical profiles. Over the years, identification method miniaturized commonly used biochemical reactions into a more convenient format and developed into a system-dependent approach that became the industry standard. The system development method relies on a set of substrates that are carefully selected for their positive and negative reactions. These patterns create metabolic profile that are compared with established database. The Microscan walkaway is an automated benchtop analyzer that uses biochemical reactions to identify bacteria down to a species level. The Microscan walkaway identification method miniaturizes commonly used biochemical reactions and can be utilized in smaller, low-resource laboratory settings. The walkaway analyzer is a fully automated instrument that incubates panels, automatically adds reagents when required, reads and interprets results, and prints results without operator intervention. Rapid fluorescent panels processed by the Microscan system provide quick and accurate identification and susceptibility results for a wide range of aerobic bacteria. Let me show you how to inoculate a panel. You will need a Renoc pipette, a panel lid, a new panel that has not been opened and was allowed to come to room temperature. You will also need a seed tray with a transfer lid, and you can see here are the individual pipettes which will rehydrate each well of our Microscan panel. This is a blood agar for a purity plate. And of course, your unknown. You will also need inoculum water and an inoculum wand. So let's get started. Let's start by making a 0.5 McFarland standard bacterial suspension. So that will be your new lid. That will ensure we have the appropriate amount of inoculum, and that is where you will be touching your colonies. So you're going to want to try and get some isolated colonies. Take your tip and touch 1, 2, 3 times. And take that piece and pull straight up, and that can be discarded. Open up your water, place tip down, and secure on tightly, and shake vigorously to ensure proper mixturing. Check the integrity of the panels and allow them to come to room temperature. A gram stain must be performed before inoculation of the panels. The panels are specific for either gram-negative or gram-positive organisms. Results may be invalid if a gram-positive is loaded on a gram-negative panel and vice versa. Make sure to place the cover on to avoid contamination. A purity plate must be made to ensure that only one organism is being tested. Using the tip from the inoculum water bottle, I streaked a clean blood agar plate. The inoculum water can now be poured into the seed tray base. Try to make sure that there are no bubbles present. I tap the corners to ensure that the lid is on securely. Now, place the re-knock on top of the transfer lid and depress the pickup levers using your thumb and fingers. Fully lift the center lever to draw the inoculum water into the transfer lid, then release hand pressure. The mechanical catch will hold the center lever in place. Pick up the re-knock with the transfer lid attached, holding it level, and position the transfer lid onto the Microscan panel. Depress the center release button. Lift straight up, put the lid back on, and depress the side levers. Make sure to dispose of the seed tray in an appropriate container. And you are ready to load onto the Microscan walk-away machine. The bacterial suspension was transferred to these panels using the Microscan re-knock inoculating device, which delivered 115 microliters of broth suspension to each well. There are different types of panels, some for identification only, and some as a combo of identification and susceptibility patterns. As you can see, there are some reactions already taking place. A label will be placed here and it will be loaded onto the Microscan walkaway in that fashion. To load it, first you need to ask for access by hitting the unlock button. You should be able to hear it, and it will go from red to green. You will open the door and make sure that there is an empty place for your panel. With the sticker facing in, the panel will be loaded and slid in just like this. I always wiggle it a little bit to make sure that it is in its place before I close it. Don't forget to lock it before you walk away, or it will alarm. Just hit the lock button again and the light will change from green to red. Don't forget to place your purity plate in an appropriate incubator. Make sure to always check your purity plate when interpreting results. Results should automatically be uploaded to the Lab Pro software. Results can be read manually using a light box.